guys, my name is Miriam Baranchek, and I'm going to talk to you guys about how cells divide. So they divide through a process called mitosis. So mitosis is going to be when one cell is going to divide and form two new cells. So with each having the same exact genetic information and amount of information, and they're identical to each other and to the parent cell. So this is an important process in the growth and replication of organisms. So we see an example of this in this tomato plant here. So you get this young tomato seedling and it grows into the plant here. And that's made possible through mitosis. And you're also going to see an example of this in how bacteria replicate and form new bacterium. So I just want to talk to you guys about the different stages by which this happens. So how does that one cell form these two different cells? So the first stage is prophase. So what's happening in the cell at this point? You're going to start to see the nuclear membrane, which surrounds all of your genetic information, break down. There's little holes that are going to form, going to start to fall apart a little bit. And then your genetic material, which is going to be your DNA, is going to start to condense. So before it does this, it kind of looks like spaghetti. But we don't want it to look like spaghetti when the cell starts to divide because it'll be prone to breaking and falling apart. So it's going to have to package itself up into tight little bundles of information, which we call chromosomes. So then the centrioles, which are these blue kind of Y-shaped looking things on my screen here, they're going to start to move towards opposite ends of the cell, and they're going to be important later on in moving stuff around in the cell and helping it to stay organized. Okay, so prometaphase. What's happening in the cell? So the nuclear membrane by this point has completely disappeared. The centrioles reach the sides of the cell and they start to send out these spindle fibers. And the spindle fibers are kind of like ropes. So they're going to reach out and attach onto like the chromosomes and sides of the cell and move and pull stuff and push stuff around. So they'll push the sides of the cell out to help it like elongate and have room for all this stuff going on inside. And then they'll also attach onto the chromosomes and pull them to where they need to be. So also during this point, you're going to start to see the chromosomes kind of moving towards the middle of the cell so that they can be readily accessed by each of those, or by the spindle fibers sent out by each of those centrioles. Okay, so metaphase, so this is the third stage out of the 5. So metaphase, what's happening? So by this point, the chromosomes have aligned in the center of the cell. They're kind of right in the middle there on this flat plane, kind of like a plate in the middle. We call it the metaphase plate. And they're being moved to that point by spindle fibers, which have attached on both sides. So each of those chromosomes is going to have 2 spindle fibers that are attached to it. So anaphase. At this point, the spindle fibers, because they're each pulling, there's 2 spindle fibers on each chromosome, they're pulling on it towards opposite ends of the cell, towards those centrioles. So this is going to result in those chromosomes kind of breaking into 2. So each of those chromosomes will divide and form 2 daughter chromosomes, and each of those daughter chromosomes will have the same identical information on each of them, so like one to the other. So in this case, like those 4 chromosomes at the top are going to be the same as the 4 chromosomes on the bottom. So that kind of helps in, like, once those 2 cells divide off, they'll each have that identical information. All right, final stage, telophase. So at this point, the daughter chromosomes have arrived at opposite ends of the cell. They've been pulled over there by those spindle fibers. The spindle fibers are going to disappear because we don't need those like ropes anymore because we already moved everything where we need it to go. The nuclear membrane is going to start to reform around those chromosomes so that they can unpack and I guess get settled in So yeah, because now that they're not being moved around, they don't need to be so tightly bundled up anymore. And we're going to want to start like replicating that DNA and processing stuff and kind of get the workings of the cell back in order. Also at this point, you're going to start to see those like cytokinesis of those two cells. So this is basically dividing of all the material inside the cell. So the one giant kind of oblong peanut-shaped cell is going to loop off and form those two daughter cells. And each of those will be identical to each other. And yeah, that's how they're formed. So thank you guys for watching. Hope you enjoyed this as much as I did. And I hope you learned a lot about mitosis. Bye.